Okay, hello everybody. I'm Brian, and this is AAMI News and Conversation. We're here at day 3 of KILMER 2025. I'm here with one of our poster session presenters, Nick Bielski. And Nick, I loved half of these posters out here, but yours jumped out at me because it's talking about genetics. I haven't seen a lot of genetics at KILMER. We're usually on the, I guess, the micro level, but we're not going deeper than that. But
Yeah.
in your case, I'm hearing about base pairs and DNA, and that excites me. So why don't you tell me a little bit about how that's relevant to sterility assurance?
Yeah, we've been really focused on sterility, but I kind of wanted to do what happens when, sterility may fail and you get these contaminants.
Hmm.
And so my focus was using next generation sequencing or NGS to identify and characterize different microbial contaminants. I mean, the theme of this year's Kilmer is next generation quality and sterility
Seems
assurance.
pretty relevant.
I figure, how can't you have next generation sequencing in the theme? It's literally in the name.
There it is. So what is next generation sequencing? What makes it different than, say, I guess what I use for 23andMe?
23andMe is actually next generation sequencing.
No kidding.
Yes. But your traditional Sanger sequencing, which is how a lot of IDs are made and how our classical sequencing happens, is you have this strain of DNA that you're doing a unique kind of PCR on, and you're sequencing up just that one strand at a time.
Mm.
So it's kind of limiting. You can only get short fragments here and there. And if you want to sequence something big, like a genome, it's going to take a lot of runs to do that, which the Human Genome Project did back in the day.
Quite enormous. I think
Uh,
that was 17 people's
yeah, it
genomes
was
all
many
combined.
years.
Quite a lot.
Yes. But next generation sequencing is kind of taking that a next step further
Hmm.
and allows you to sequence a bunch of things in parallel. And it's that parallel sequencing that allows you to really get the volume and really help speed up the process.
I know there's a lot of data to work with, but I guess in parallel, we're going to save a ton of time and money.
Oh yeah, but then you get a lot of data, and that's going to take a lot of processing. So you get some pretty big files.
So that's what I'm wondering. Yeah, now we have that. And I think this was something that was mentioned when the Human Genome Project first started. They said, we have big data. It's going to take us a long time to figure out, make sense of it all.
Yes.
So how do you make sense of what you're getting?
I try my best. To be honest, I mean, what I presented on is kind of suggestions for how, this community can take that data and really use it for something meaningful.
Gotcha.
But that's just suggestions. I think this technology has a lot of opportunity to go further.
Mm.
There's a lot of data, like you said, and parsing it can take a lot of time and really trying to figure out what maybe unexpected things you can get from it and how it
Yeah.
can help you. So I really focused on, how you can ID things with NGS and how you can take that further and compare different genomes. So if you want to see how closely related two strains are, you can look across the entire genome, or even just searching that genome for things like antimicrobial resistant genes or any kind of virulence factors. See if you can find any, precursor identifiers, like if there may be some concerning phenotype.
So I was going to ask you, what are you looking for differentiator wise? Because you're talking about, essentially different strains.
Hmm.
And I think you nailed it. You said antimicrobial resistance. That's going to be, if it
It's important.
fails, that's the big why. Why did it fail,
Hmm.
the sterilization process? So
Yeah.
there we are. Is that it?
Yeah, I mean, we can't tell you for sure that
Mhm.
it's going to have that. There's a lot of factors going from your gene to your protein to
Yeah.
your phenotype. But, if it doesn't have the gene, it's not likely to have it. If it does have the gene, it does give you some more cause for concern. And so
Mhm.
it's definitely something to look into. And, comparing the strains, typically you would do that with just a select group of genes that are established and published for an organism. But if you can sequence entire genomes, you don't need a set group
Yeah.
of genes. You can actually look across any sequence variant you see and use that to do your comparisons.
So your poster is essentially kind of a call to action. Here's something cool. Here's what we should start looking into more research to come kind of deal. But what are you hoping to see as like the final result? Let's say all the in 10 years, 20 years, all that research is complete. You've been collaborating with people
Hmm.
and we have a better sense of how to go through this process. Give me a use case. What at the end of the day are we going to see as a application of this?
I mean, if you want to, go around your facility and take swabs everywhere, you don't have to take plate samples.
Hmm.
You can just take a swab, you can extract that DNA, and you can identify any possible contaminant. It was mentioned in an earlier session how, we're reliant on things to grow on plates. So if something's not culturable, you're not going to see it. That's not something in NGS if you don't have to culture it. You can take your samples and you can just put them into the workflow. And I think you can do tracking. If you see something show up in one room, you can have a sample from another, and you can have consistent monitoring of your environment with NGS, constantly looking at the different organisms. And if you do find something, you could take all those strains, sequence their entire genomes, and compare them that way. And another really interesting thing to think about is if you're doing a lot of this, you could kind of build your database for your factory of common microbes that you see and have their entire genomes. So if something new pops up or something else pops up, you can compare it back to your personal database and see, is this something you've seen before? Is it something new? Where might it be coming from? And it might really help you in some of these contamination control investigations.
That is very fascinating. I was like, this is going to inform risk assessment, things
Hmm.
like that. That's very cool. All right, so awesome research. I'm looking forward to seeing more about this in years to come. But you're also brand new to Kilmer. This is your
Yep.
first conference. What did you think?
It's been great. I am newer to the sterility field as well.
Mhm.
So it's been really nice to see kind of a lot of what goes on prior to finding a contaminant and how much effort people are putting in. I mean, coming from, someone who doesn't really think about this stuff or who never really did, I think it's so surprising and it's actually amazing seeing how much people care about something and how much they put in effort to sterility. And it's not something I ever really considered,
Yeah.
like all the different factors that come to play, but how important they are for human health. And it's really just amazing seeing so many people who've been dedicated to this and who are working tirelessly behind the scenes that you don't even really think about, just through your regular everyday life,
Yeah.
but has such an impact.
So you're going to be here in 4 years? Next one?
Certainly hope so.
All right. I look forward to seeing you then. Thanks again, Nick. Enjoy the rest of Kilmer and congratulations.
Thank you.
All right.